The story you have depicted here is a visual summary of the Brenner, Jakob, and Messelson experiment that was done in 1960. They sought to further study and visualize the process of taking DNA and making proteins. What they did was they grew bacteria in heavy tagged radioactive carbon and nitrogen to obtain identifiable ribosomes. These were then added to different radioactively tagged phosphorus solutions and bacteriophages, which would allow the bacteriophage to introduce new genetic content. The assumption was that they would be able to see the DNA protein conversion mid-act because the amino acid building blocks were excluded. After spinning the solutions down, they found no DNA at all, but something else entirely, a biological genetic middleman. This middleman is ribonucleic acid, or RNA. The monomers for RNA are very similar to DNA. They're nucleotides, adenine, cytosine, guanine. The big difference here, though, is we use uracil instead of thymine. Another interesting thing is the type of sugar. DNA has a deoxygenated ribose, and RNA has an oxygenated ribose. Also, too, the strands. DNA is naturally double-stranded, while RNA is single-stranded. Be aware of these key differences. And so from this experimentation, we come to understand the central dogma of molecular biology. This is the general flow of genetic information. Know this flow. When DNA is made into more DNA, we call that process replication. That DNA is typically converted to RNA, leaving the nucleus, in a process called transcription. And that RNA then becomes translated into proteins. It is the proteins that are the doers, movers, and shakers of the cell. Now, there are specialized enzymes that can allow the reverse of information, reverse transcription, if you will. But generally speaking, the flow is DNA to RNA to protein. Let's talk a little bit about replication, the synthesis of DNA into more DNA. It starts with double-stranded DNA. It ends with double-stranded DNA. Transcription then takes the double-stranded DNA and synthesizes a single-stranded RNA message that can leave the nucleus, go out into the cell, and be expressed. Spread the news. Translation is what happens to that messenger RNA, and it is the synthesis of a polypeptide proteins chain that is able to do stuff, to actualize the instructions. As you can imagine, the process of moving DNA to RNA to protein, replication, transcription, and translation all require specialized enzymes and components to facilitate these tasks. It's interesting to note that in the beginning, early life, our fossil record shows that RNA was the first molecule of heredity, that specialized RNA sequences called ribozymes were able to be both RNA and protein-like sequences. But over time, that single-stranded nature of RNA is rather unstable and easily damaged. So over time, evolution led to DNA being our stable genetic information storage molecule. And so recognize the specialization. DNA stores genetic information. RNA sends messages, translates it, and proteins do the work. Why can't DNA just go straight to protein? Why did evolution favor this specialization? That RNA intermediate provides protection for the DNA and allows more copies of protein to be made simultaneously, since many RNA transcripts can be made from one gene, and each gene transcript can be translated repeatedly over and over again. The summary is much more accessible than the entire book. Recall that prokaryotes are simple, single-celled organisms. There is no nucleus, therefore no separation of space or time from DNA and RNA. This creates an all-or-nothing pathway. The minute transcription has begun, translation is ready to go. Both can occur simultaneously. This allows this process to be quicker, but at a cost. There's less regulation, and therefore it's error-prone. Eukaryotes, on the other hand, have a nucleus. They also have many enzymes that can help along the way. So that nuclear envelope separates the transcription process from translation, creating regulatory checkpoints. RNA processing also allows us to modify early mRNAs, creating timing controls. All those extra enzymes and these checks and balances allow us to have quick efficiency without the error that our prokaryotic counterparts have. And those processing allow the products to live longer than the prokaryotic counterparts. So the value of being a eukaryote is increased efficiency and longevity. Transcription is sometimes referred to as RNA synthesis. It's the process of making snippets of information from the DNA template. There are 3 types of RNA, and each one has its own specialized function. mRNA is the messenger RNA. This is used to communicate and pass genetic information from DNA to protein. rRNA, the R stands for ribosomal, and this type of RNA can fold upon itself and, like its ribozyme ancestors, engage in enzymatic processes. This actually forms the ribosome, which participates in translation. tRNA, the T stands for transfer. Like the ribozyme ancestors, this RNA sequence will fold upon itself and become an enzyme doer that participates in translation by moving the amino acid to the appropriate nucleotide codon. The process of transcription can be broken down into 3 simple stages: initiation, elongation, termination. Transcription begins at a sequence known as the TATA box. The TATA box is a specialized area of the genetic code that's rich with adenine and thymine nucleotides. You might recall that these nucleotides bond together with only 2 hydrogen bonds, as opposed to cytosine and guanine that bond with 3. This means that that segment is easier to separate, making it more accessible to be read. There are enzymes and other proteins known as transcription factors that will bind to this promoter region to help stabilize the DNA and unwind it to make it more accessible. But the key player here is RNA polymerase. It synthesizes the RNA by following along the DNA strand and adding the complementary base pairs. So here we can see that in visual diagram. We can see our TATA-rich promoter box and the transcription factors binding to that region. And just where the start point is, we see our bubble separate those 2 strands of DNA and an RNA transcript being formed, moving along in the 5' to 3' reading frame. This brings us to elongation. The RNA polymerase will move along the DNA, going 5' to 3', untwisting the DNA helix, making 10 to 20 bases accessible at a time. And it will add a complementary base based upon the DNA template at a rate of about 40 nucleotides per second in eukaryotes, creating the single-stranded, newly made RNA transcript. How does it know it's done? In bacteria, polymerase will stop at the end of a terminator sequence, and the mRNA is ready to go right then and there. It's released and able to be translated or folded up and utilized as a tRNA or rRNA immediately. In eukaryotes, however, the RNA polymerase will continue for hundreds of nucleotides past the termination sequence until it reaches a secondary encode. In eukaryotes, it needs to have confirmation, a double end, a "are you sure you're sure" stop code, before the pre-mRNA is cleaved from the growing RNA chain. I call it a pre-messenger RNA because it's not yet ready to be translated or fold up and act as an RNA, ribosomal or transfer RNA. It needs to have some modifications. Translation is often referred to as protein synthesis. This is the process of reading the mRNA message and translating the nucleotides into corresponding amino acids to create proteins that can act as transporters, communication signals, enzymes, or provide structural integrity and strength. The key player of translation is the ribosome. The small subunit of the ribosome will bind to the mRNA binding site, recognizing the start codon. The large subunit of the ribosome will bind on top of this, sandwiching the mRNA transcript. Another key player is tRNA, the transfer RNA. Each tRNA carries a specific amino acid and has a specific anticodon on the other end. And so when the units of the ribosome come together, the transfer RNA can bind complementary to the RNA transcript and deliver the appropriate amino acid. Once it does, the tRNA will leave, and an amino acyl tRNA synthetase enzyme will help match it back to its correct amino acid so it can be reused and redeliver new information. The 3 stages of translation are very similar. There's an initiation phase, an elongation phase, and termination phase. All 3 stages require protein factors that will aid in the process. Translation begins when the small ribosome subunit and its special initiating tRNA find a start codon, a special unique nucleotide sequence of AUG. Once they sandwich, additional proteins called initiation factors will help bring the large subunit to complete this complex. During elongation, the process proceeds in a 5' to 3' direction. Binding sites within the ribosome, an A, P, or E site, regulate the addition of amino acids one by one. This involves elongation factors, codon recognition with complementary pairing, a peptide bond being formed, and then the shifting of the ribosome, translocation down the transcript. In each of those sites, something unique is happening. The A site stands for attachment. This is where the amino acyl tRNA will bind to deliver the appropriate amino acid. It has to match the appropriate codon and anticodon pair. The P site is where the peptide bond will form, delivering and depositing the correct amino acid. The E site is when the tRNA, now without its amino acid, will leave to come back again after it has been fused with a amino acyl tRNA synthetase. Know what's happening in each site. A, it attaches. Then there's translocation. The ribosome shifts. And what was in A suddenly is now in P. And in the P spot, it will peptide bond to the growing chain. And then there's translocation. What was in P now moves to the E site. But now it's lacking its amino acid. And so it's kicked. E for exit. This will continue until a stop codon in the mRNA reaches the A site. At this point in time, a release factor, which is really a water molecule, is added. But because this is not held together by the same bonds, it stimulates the release and freeing of the polypeptide chain. Part of the efficiency of translation comes from the fact that any number of ribosomes can be translating a single message RNA simultaneously. This enables the cell to make many copies very quickly. So here we can see it in both graphical representation as well as in an electron scanning micrograph.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Recall that a mutation is a change to the genetic material, and this can occur within a cell or virus. Mutations can affect one nucleotide or several nucleotides, and sometimes that change can affect the overall protein structure. Given that form dictates function, it can have lasting downstream effects. On the bottom, we see an example of where a single nucleotide change in the DNA template can lead to the production of abnormal proteins. Healthy red blood cells into sickle-shaped red blood cells, unable to carry oxygen. Point mutations are caused by nucleotide pair substitutions, where one nucleotide is replaced by another one. The results can be silent, missense, or nonsense mutations. Know the difference between these 3. A silent mutation changes the sequence of the genetic information, but it's considered silent and not harmful because the overall amino acid has not changed. This is due to redundancy in the genetic code. Sometimes, however, that substitution will result in a new amino acid being added. This is termed a missense mutation. If the new amino acid happens to be a stop codon, you get a nonsense mutation. Nonsense mutations nearly always lead to a non-functional protein that will be degraded and reused, but can be problematic because it has utilized resources and delayed the replenishment of a much-needed protein. Taking a look at each of these examples now in visual form. Here we can see a single nucleotide substitution, an A instead of a G, which then leads to a U instead of a C being incorporated. But because the wild type and the mutated strand both add a glycine, it is a silent mutation. The overall protein transcript has not been altered. In this example, a thymine has replaced a cytosine, and that leads to the mRNA transcript having an A instead of a G. This codes AGC versus GGC, and that changes which amino acid was added, a serine versus a glycine. This is a missense mutation. In this last example, adenine replaced thymine, leading to a uracil in the mRNA transcript, and UGA codes for a stop. This sequence is utter nonsense because it literally was one amino acid and ended instead of continuing on the code and adding the lysine, the phenylalanine, and glycine beyond. The other type of mutation is the result of insertions or deletions. Any additions or losses of nucleotide pairs in a gene may alter the reading frame, producing a frame shift mutation. This is missense on high. It doesn't just change one amino acid, but every amino acid after the fact. The example here is the fat cat stat, common English words that follow the 3-letter codon pattern. Let's say we delete the T. There is no space saving amongst codons. What happens is everything shifts down. And so then we only get that first codon that made sense, the. After that, utter gibberish, with every amino acid after the addition or deletion being altered in some way, shape, or form. That is, unless the insertion or deletion occurs in multiples of 3. So here we see that depicted. A single A deletion results in a U deletion, And that changed phenylalanine to leucine, but more importantly, glycine to alanine, and so on and so forth, on and on and on. Where do these mutations come from? Many of them are spontaneous. They just happen, errors during DNA replication, recombination, or repair. But sometimes they can occur more frequently due to mutagens, physical or chemical agents that damage our DNA. The genetic code is nearly universal, shared by the simplest bacteria to the most complex animals. We know this because we can move genes from one animal to another, and it will still express. It can be inserted, and our current machinery can process that information. The diagram here shows bioluminescence from bacteria being placed in both a plant and an animal. Genes, nucleotides, they're just bits of information. It's where that information is expressed that makes all the difference. So what is the code? The code is really the interpretation of codons to amino acids. A codon is 3 non-overlapping nucleotides. And because we have 4 nucleotides, A, U, C, and G in RNA, they give rise to 20 total amino acids. 20 essential amino acids that we absolutely must have to function properly. Each codon is specific. A codon will always code for a particular amino acid. CUU is always leucine. But there's something called redundancy here, also known as degeneracy. Notice in the third base of the codon, it doesn't matter if it's a U, a C, an A, or a G. All 4 of these variations still code for that same particular amino acid. What we see is that in the genetic code, about 50% of the time, the third base doesn't matter. But there are several code words that ultimately have the same meaning. And this is due to a process, a phenomenon known as wobble fit, that the third base doesn't sit as well during our complementary base pairing of the codon and anticodon. And so over time, the ribosome has evolved its own set of rules, and it will almost always exclude or ignore the third base in the anticodon pair to prevent massive missense mutations occurring. And so we see in this table showing the codons and their appropriate amino acid codes that there is a lot of redundancy, multiple ways in which you can code for the same amino acid.